Hello everyone. Today we'll learn about restriction endonuclease and its type. Restriction endonuclease or restriction enzymes are important tool used in recombinant DNA technology. It is capable of making cuts the desired DNA at specific sites. It is also called molecular scissors or genetical scissors. Restriction endonucleases were discovered and characterized by Arbor, Smith, and Nathans, and they won the Nobel Prize for Physiology or Medicine in 1978. Where restriction enzyme is found? It is found in almost every microorganism, but more in bacteria. In bacteria, it acts as chemical weapon against viruses. It restricts the multiplication of viruses, thus called restriction enzymes. Now come to the nomenclature. Each enzyme is named by 3 or 4 letter abbreviation. They are named according to the name of organism from which they derived. The first letter of the genus together with first 2 letter of a species name of the organism. After that, the first letter of strain in capital. They all together form the enzyme name. Roman number at the end of enzyme name indicates the order of their discovery. Examples: Eco R1, Hen 3 and Balm H1. You can see here the nomenclature of restriction enzymes. These are few examples of restriction endonuclease with their origin and recognition sites. Types of cleavage. Restriction endonuclease make 2 types of cleavage. First, blunt end fragments, and second, sticky or cohesive ended fragment. The blunt end DNA fragments are formed when the restriction enzymes act on the line of symmetry, that is, they cleave both DNA strands precisely at opposite point. And cohesive end fragments, the molecules are produced when the enzyme acts around the line of symmetry, that is, 2 strands are not cut on opposite points and cuts are staggered. The restriction endonuclease often recognize palindromic sequences. The palindromic sequences are inverted repetitive sequences which are symmetrical. Palindromic sequences have complementary sequences at the 2 ends of a single strand. We can read palindromic sequences same as forward and backward, just like madam. Important features of restriction endonuclease. The important features of restriction endonuclease are: first, these enzymes make cuts in palindromic sequences, each consisting of 4 to 6 complementary bases. Second, the breaks are usually staggered cuts forming cohesive strands. And third, the enzyme yields DNA fragments with complementary ends. Now come to types of restriction endonuclease. Restriction endonuclease is of 4 types. Restriction endonuclease 1, 2, 3 and restriction endonuclease 4. Restriction endonuclease 1. In this system, same enzyme possesses both activities, that is restriction and modification. Restriction means cleavage and modification means methylation activity. It is bifunctional. It cuts both strands at a non-specific location. ATP, Mg2 plus and S-adenosine methionine are the cofactors of restriction endonuclease 1. It possesses two recognition sites. The example of restriction endonuclease 1 are ECO K12, ECO B, ECO A1. Now restriction endonuclease 2. Restriction endonuclease 2 have separate enzymes for modification and restriction. They are independent of its methylation and cut DNA at specific site. Other cleave DNA randomly. They cut DNA at specific sites use palindromic recognition sites, that is 4 to 6 BP in length. It has single recognition site. It has only one cofactor, that is Mg2 plus. The first type 2 enzyme is HIND2, which was isolated in 1970. Type 2 enzymes are stable and most important for cloning purpose. The examples are EcoR1, BAMH1, HIND3, etc. Restriction endonuclease 3. It has same enzymes for both restriction and modification, just like our restriction endonuclease 1. They cut DNA up to 20 to 30 BP away from recognition site. Mg2 plus and S-adenosine methionine are cofactors. Examples are EcoP1, EcoP15, HIM3, etc. Restriction endonuclease 4. This kind of restriction endonuclease cleave only methylated DNA. It shows weak specificity, MC, RBC and MRR. These are the comparison between three types of restriction endonuclease, that is 1, 2 and 3. Now come to the application. The application of restriction endonuclease are as follows. It is used in gene cloning and protein sequencing. Second, it is used in preparation of restriction map, in restriction fragment length polymorphism. Restriction endonuclease used in the formation of recombinant DNA, and it is used in nucleotide sequencing. Thank you.